Hi everyone, and welcome to Chapter 42, which focuses on the immune system. Now, I will warn you, this is one of those chapters where if you try and look up this information online or reading some textbooks, they can get really complicated and overly dilute where, they kind of miss the main points. So I try to instead make this chapter as painless as possible and kind of focus on the key concepts. Now, the first thing to point out is the difference between your innate and adaptive immune responses, meaning your immune system has two different versions to help you in everyday life, to help protect you. Now, when you look at these terms, innate, in the English language, anything innate means that you usually don't even notice it because you're born with it, okay? Innate is anything you're born with. So your innate immunity is your natural, born with immunity. It is general. It is non-specific, meaning it is there to protect you from any invaders in your body very quickly without taking the time to identify them and tailor its attack. Instead, it is meant to block and attack anything that comes its way and not kind of, try and figure out who the invader is. Adaptive, on the other hand, if you think of adapt and adaptions, you think of long time evolution, changing, growing with time. And that's what your adaptive immune response is. It's your immune response or your immunity that changes throughout your lifetime. Okay, it's constantly evolving, because this is the very specific immune response, okay? This is where your immune system takes the time to identify the exact pathogen that is invading your body. It develops specialized targeting attacks, and it creates memory cells so that in the future, when you encounter that same pathogen again, your body can quickly defeat it because it's already got the protective cells in your body. So when you hear that you can't get sick twice from the same infection, it's due to your adaptive immunity. Now, it's not that you can't get sick twice. Let me just clarify that for you, because I know students hear that all the time. What it means is that same pathogen can enter your body again a second time, but you usually won't notice it and you won't develop the extensive symptoms that you got the first time because your body already has the memory cells, and it doesn't have to take time to make all of the adaptive cells that you're going to see later in this chapter. Okay, because this is a very specific response and takes time to activate and differentiate different cells, it is very slow the first time. Now we're going to go through both types of immunity, the first one being your innate immunity, the one that you're born with. There are two types of innate immunity. There are barriers, which are your first line of defense, and then there are additional mechanisms that will help kind of further back up your immune system. Now, I want to emphasize with the barriers, when you think of barriers on your body, you'll think of things that are blocking the entry of pathogens. Now, I want you to write down that barriers can be mechanical, chemical, or microbial, meaning your normal flora, the microbes or the bacteria that live within and on your body. Okay, so your barriers, they're not just always physical. Barriers can also be chemical or microbes, but good microbes. Now, just to give you some examples of barriers, the first one, which is a big old barrier, is your skin, which is actually your largest organ, because remember, the skin is made up of many different types of tissues working together to help protect you. Now, you already know that the skin is made up of the outer portion, which is called the epidermis, which is very tight epithelial cells and keratin. And then you have the inner portion of connective tissues, that's the dermis. Now, your skin protects you in a lot of different ways, but one of the big things I want to point out is your skin helps you get protected from desiccation, okay? Desiccation is a very fancy way of saying drying out, okay? So your skin is not only a physical protective barrier around all of your other organs, but it also helps protect the hydration within your body. You're also going to see in later slides that your skin helps protect you by also having hair all over it, by having oil, which we call sebum, and by having things like sweat and altered pH that makes the skin unfavorable to some, bad guy pathogens that might try to attack you. You also have as barriers your mucous membranes. And I know a lot of people, when, they see mucus, they see snot running out your nose, or they hack up or cough up some, globs of yellow or brown or clear, gunky mucus. They think, ew, that's disgusting. I don't want that around me. Well, your mucus is actually part of your immune system. Your mucus lines all of your very important tracts, such as your respiratory tract, your digestive tract, all the ones that I listed out in the first bullet point. And the benefit of this gross, globby, sticky kind of mucus is the fact that it's what we call viscous, V-I-S-C-O-U-S, which is a fancy way of saying it's sticky, right? It's sticky like honey. That's beneficial because it traps microbes. It also, by lining these tracts, helps prevent drying out of those tracts as well. Kind of like we said, the skin helps protect you from drying out. Your mucus further helps prevent drying. But the big point I want to make about mucus is the fact that pathogens or microbes, meaning bacteria, viruses, fungi, they can get stuck in this sticky mucus, and then what do? You go, you cough it out, right? Or you blow your nose and you get it out of your body. So it's the benefit of trapping the microbe and then forcing it out of your body. You also have the epiglottis, and the epiglottis we've mentioned before in the digestive tract because that is a flap at the back of your throat. It protects your respiratory system. That's why it's considered part of the immune system as a barrier to your respiratory system, because you do not want pathogens or anything getting into your lungs, into your respiratory tract. Okay? You know that those end up causing a lot of complications. So your epiglottis helps as a flap protecting, blocking your respiratory tract, blocking the top of your trachea. And this is why I said in the previous lessons that you should never talk while you're eating, okay? It's because the epiglottis can open up with the vibrations of your larynx, of your speaking, and food can go into your respiratory tract and you can choke, okay? So this little flap here is your epiglottis. Then you have some very important chemical barriers. Now we call the lacrimal apparatus and saliva as both physical and chemical protection. The reason why we consider them physical protection is that your lacrimal apparatus, which is a fancy way of saying your tears, okay, your tears and your saliva, they're liquid. And anything on your body or in your body that is liquid has the physical protective function of being able to flush things away or wash away pathogens or microbes. Now, in addition to being able to wash away physically pathogens or microbes, both your tears and your saliva have very valuable chemicals that can protect you. Okay, so tears, for instance, they contain 3 enzymes, I want you to know. They contain IgA, and you can circle the A of IgA and remind yourself that IgA blocks microbe attachment. Okay, so circle the A of attachment and the A of IgA, and it reminds you that IgA blocks microbes from being able to attach to your tissues and cells. It also helps neutralize some toxins that those microbes, those pathogens, might secrete. Next, you have lysozyme, and you'll notice that IgA and lysozyme are both in tears and saliva. So saliva will have these same two guys, IgA blocking attachment and lysozyme destroying the cell walls of pathogens. They destroy what's called the peptidoglycan. Remember, we mentioned that's an important carbohydrate for bacteria. So lysozyme, if you think lyse, like cell lysis, lysozyme can break up bacterial cell walls. Then tears and saliva have a unique third enzyme. For tears, they have lipocalin. Okay, lipocalin, I see that I in lipocalin, and I think iron. What lipocalin does is it binds iron, which is an important cofactor for pathogens. And cofactor, you know, means helps activate enzymes. So if lipocalin binds up the microbe's iron, it blocks the microbe or the pathogen's metabolism. Okay, so lipocalin binds iron and blocks metabolism of pathogens. Whereas saliva, the third enzyme, which is unique to it, is histatin. I want you to circle star, highlight that one. That one is an antifungal. So that's why fungal infections are rather rare in your mouth, because you have a nice antifungal in there. So anytime you think of tears and saliva, please remember they are part of your innate immunity. So basically the way I think of it is, anytime I make someone cry, such as my students on exams, I'm doing you a favor. I am protecting you with your immune system. So you're welcome. This slide is just to list out some more barriers, because we can't go through every single barrier in your body. But some of the other important ones that you might encounter on a daily basis are your gastric juices, and you will notice they have a very acidic pH. So do urine and vaginal secretions. They also have a very acidic pH, which means all of those have the immunity benefit of basically making conditions unfavorable for pathogens. You know, a lot of bacteria do not like very strong pHs, very strongly acidic locations. You also have earwax. That is a barrier of your innate immunity, because just like with mucus, earwax is sticky. Okay, I know you don't want to think about it, but earwax is sticky, which means that it helps to trap some pathogens, and then you end up removing it from your body. You also have peristalsis and vomiting, meaning those rhythmic contractions in your digestive system, then pushing vomit up and out, or defecation, meaning poop, and diarrhea. Now, with both of these, I know you may think that they're disgusting, but they're actually part of your immune system trying to protect you. And think about it. I know you don't want to think about vomit and diarrhea very long, but think about it. What are they trying to do? They're trying to quickly get out the toxins or pathogens that have come into your body. Okay, and that's why when you have any kind of vomiting or diarrhea, the first time or the second time that you have it, you should not be quick to take any medication to stop it, because you want to get those toxins and those pathogens out. Now, when does vomiting or diarrhea become a problem? If it occurs too much, and then dehydration can become a risk. Okay, but you want to kind of hold off on medications, because in fact, when I teach microbiology, I show students there are a lot of gut infections where taking an antidiarrheal actually allows that pathogen to then stick and attach within your digestive tract, and it becomes much more dangerous and harmful to you. Okay, so if you can avoid it, you know, let yourself have a little diarrhea and vomiting, and then just be careful to make sure to replenish your electrolytes and water. Now, in addition to all those barriers, your innate, or you're born with it, immunity also has molecules in cells to be a second line of defense for any pathogens that make it across the first barriers. Now, keep in mind, as we talk about the innate immunity, again, these barriers and these molecules like toll-like receptors and cytokines, all of these are basically trying to get rid of or block any pathogen. It is not taking the time to learn who they are. This is all general, non-specific. So please circle innate immunity and keep reminding yourself that it is general, non-specific barriers. So it is quick. As soon as it encounters a pathogen, quick. Now, what toll-like receptors and cytokines do is both of these are proteins or chemicals involved in triggering your immune responses. Okay? They help attract your white blood cells and your immune cells to help fight the pathogen. Okay? The key here is they trigger immune responses to help get rid of the pathogen. Now, when I say your white blood cells, I mean your various leukocytes or cells that are trying to protect your body. So here I listed out the many cells of your immune system, okay, trying to protect you, and we're going to go one by one through each of them to kind of look at the key points about each one. The first one to mention are your neutrophils. And notice that your neutrophils and many of your other white blood cells, including your eosinophils, are phagocytic. Okay, phagocytic means that they engulf pathogens. Okay, they can cell eat other cells, meaning they can engulf and break down pathogen cells. And again, pathogens mean the bacteria or the viruses, the bad guys attacking your body. You also have basophils. I want you to circle, star, highlight basophils and histamine, okay? Because when you think of histamine, what do you think of? What kind of reaction? You think of allergies, right? Antihistamines are what you take to fight allergies. Well, basophils, okay, they secrete histamine, okay? They occur especially during allergies, which is why you have so much, histamine and inflammation during allergies. And what's important about histamine, the reason why these white blood cells release histamine, is it is a vasodilator. So I want you to circle histamine and write down that it is a vasodilator. And what does a vasodilator do? It opens or expands your blood vessels, which means it's promoting blood flow. Why is that important in your immune system? Well, if there's more blood flowing at that location, well, that means more white blood cells, more immune cells to fight the infection there, to fight the pathogen that is at that location. So that's why you see histamine come up a lot in immune reactions. Now, the downside to this is that histamine, you'll feel a lot of inflammation in that immune response, even though, your body is trying to protect you and help you with that, but it may end up making you temporarily not feel as well. Okay? You also have eosinophils, and I want you to circle, star, highlight that eosinophils are primarily for parasitic and helminth infections. And next to helminth, write worms. Helminths are worm infections. So for instance, if you ever get hookworm, okay, they like to burrow into your feet, which is why you shouldn't walk on sand or out in your front yard on dirt, anywhere where stray cats, stray animals could have been walking around and pooping, because you tend to get worm infections that way. So eosinophils help protect you and fight parasitic and worm infections, helminth infections, by producing toxic enzymes that help break down those particular pathogens. You also have monocytes, which are ultimately going to become macrophages. And for macrophages, I want you to write that these are the most phagocytic of all white blood cells. So circle macrophages and write most phagocytic of all white blood cells. Okay, that means they really like eating up pathogens and breaking them down, digesting them. They're also one of the antigen presenting cells in adaptive immunity that we're going to talk about in a couple of minutes. And we'll talk about 3 different antigen presenting cells, okay? But for now, you don't have to worry about those. Another one that we will mention as well with antigen presentation is dendritic cells. But again, for now, in innate immunity, you do not have to worry about that. The next cells are mast cells. And mast cells, they basically, when we say they produce chemical mediators, what that means is they produce chemicals that will help fight infections in your body. And the 3 that I want you to know are histamine, heparin, and chymase. Okay, histamine, heparin, and chymase. Histamine, we've already mentioned. The significant point about that is it's a vasodilator, meaning it opens up your blood vessels and helps increase blood flow to the infection site. Heparin, which I'm sure you might have heard, especially with grandparents, a lot of grandparents take heparin. Heparin is an anticoagulant. And if it's anticoagulation, what do we usually call those kinds of medications or chemicals? They're blood thinners. So just like with histamine, the idea is to get as much blood flow as possible. So not only do you have vasodilators opening up the blood vessels, you also have heparin now as an anticoagulant to thin out the blood, as a blood thinner. Then you have chymase, which is a protease, meaning it helps break down proteins of the pathogen. Okay, it helps protect you that way. The last ones on the slide to mention are natural killer cells, and that's this little guy here. And your natural killer cells are important because they release what's called cytotoxic proteins. And in biology, you always want to break down the term. So cyto, so I'm going to highlight it over here. Cyto in cytotoxic means cell. Toxic, you know, toxic destroys. So cytotoxic proteins are proteins that destroy cells, which are why they're called natural killer cells. They destroy other cells. Specifically, they tend to destroy tumor cells and any of your own cells that have been too badly infected to recover. So it wants to destroy your own cells just to help protect you from the pathogens that have infected them. Okay? So here I wrote out the 3 chemical mediators that I mentioned in the previous slide, produced by mast cells, just in case you missed what I was saying or was unsure of any of the spelling. Now, when we talk about innate immunity, I want you to know the 5 mechanisms that are involved in your innate natural immunity. You are born with immunity. Again, all of these are non-specific. They're targeting any bad guys. The first one is phagocytosis, and you're already experts at that term. It's basically cell eating. It means that white blood cells, and what did we say the most phagocytic blood cell is? Macrophage. So a macrophage or any other phagocytic white blood cell will engulf a pathogen. So in this case, a bacteria. It basically binds it with a little special receptor, like the toll-like receptors we mentioned earlier. So the phagocyte or macrophage will engulf that bacteria, and what's inside the white blood cell? There are enzymes from lysosomes in there, and you already know lysosomes tend to have enzymes that can break down that bacteria. And then what that white blood cell does is it will take some of that broken up cell to use in its own cell, basically scavenge any of the valuable parts, and then excrete the waste because it's basically cell eating. So it takes in the bacteria, it digests it, and then it poops out the rest. Okay? So that's very beneficial to you. Your innate immunity also has inflammation, which we just mentioned a minute ago. Now, I know inflammation can be very annoying and uncomfortable. It involves redness, swelling, pain, and heat, but it's actually trying to protect you because, again, the reason you see inflammation is because of the changes in the blood vessel, specifically vasodilation. Again, a lot of this is due to the release of chemicals such as histamine. Now, when you have that inflammation, the benefit is that with that vasodilation, you get increased blood flow. So you get more blood volume in that area to have more white blood cells protecting you and fighting the pathogen. So it's a good thing for you because it means more white blood cells in that location. You also have fever as part of the PIFKI, I call it, the PIFKI innate immune responses. So we covered phagocytosis, inflammation, and now fever. Now, fever basically means that your body raises the temperature, okay? It ups your natural thermostat, increases the temperature, and that's beneficial for a lot of reasons because think about it. First of all, if you're now at a hotter temperature than when that pathogen came in, well, that's going to inhibit bacterial growth. A lot of pathogens have a particular temperature they like, such as your normal body temperature, and if you raise the thermostat, they're going to be uncomfortable. It can affect their growth. It can even inactivate a lot of their toxins because it denatures them. Higher temperatures denature proteins such as the bacterial toxins. Also, if any of you have taken chemistry classes, by increasing the temperature, you're also increasing the speed of reactions in your body. So that helps defense because now you have quicker immune reactions going on because higher temperature increases the speed of reactions. Furthermore, and most important, I think, is the fact that when you have a fever, think about it. If you just have a regular old cold and you don't have a fever, what do? You still go to school, you still go to work, you might go to the gym or supermarket. If you get a fever, what do you usually do? You stay home and rest. And ultimately, that's the best thing for your body when it's fighting an infection. So a fever has that extra benefit of keeping or forcing you to rest and to heal. Okay, so some of the benefits we mentioned, it inhibits bacterial growth. The fever inactivates bacterial toxins or denatures bacterial toxins. It increases your body reactions, and it keeps you resting. Now, everyone's heard of phagocytosis, inflammation, and fever, but the last two parts of innate immunity students usually have not heard of. The first one is the complement system, and it's not as friendly or polite as it sounds. Now, again, in an upper level course, we'd go through all of the different pathways involved here. But for our purposes right now, in general bio, you just have to know that the complement system, what it does is it's a series of proteins, a whole bunch of proteins that physically destroy the bacteria by drilling holes in the bacterial cells. Okay, so I highlighted it in blue. I want you to emphasize that the complement system is proteins that will drill holes in the pathogens, and that ends up killing the pathogen. The last one I want you to know is interferon, and as soon as you see interferon, all I want you to think is it fights viruses. Okay, so all of the other parts of innate immunity, they can protect you against any pathogen, and a lot of times it's bacteria in most cases. Interferon is specific to help you fight viral infections. And what it does is it's basically a mechanism that will degrade the virus's mRNA, which is an important genomic material in that virus. Okay, So when you think of interferon, you don't have to know the mechanism that it works. I just want you to know that it is part of innate immunity to fight viral infections, viruses. And now we move on to adaptive immunity. So again, innate is the quick, general, blocking kind of immunity. And then adaptive immunity now is your more specific, long-term immunity that changes and grows with you throughout life. Now, there are two ways that you can get adaptive immunity. One is by fighting off infections that you naturally catch, and the other way is through vaccinations. So vaccinations are for adaptive immunity. And it makes sense because we said adaptive immunity involves memory, so you don't get sick again. And that's what vaccines are trying to do for you as well. Now, when you think of adaptive immunity, I want you to automatically think of humoral and cellular immunity. Okay, so when you see humoral and cellular, make note that that is not innate immunity, that is only adaptive immunity. Now, we break those down further so that anytime you hear humoral immunity, I want you to remember that that involves B cells and antibodies, and the B cells are what will produce those antibodies once they differentiate. We'll talk about that in a minute. Then cellular immunity, I want you to think T cells. Okay, so humoral is B cells and cellular is T cells. Now in adaptive immunity, because it is highly specific, it involves what's called antigen presentation. And you already know antigens, just like in your blood typing that we talked about, antigens are name tags, okay? They're proteins on the surface of a cell that basically tells your body who that is, okay? Now, in the case of fighting an infection with adaptive immunity, you have 3 types of antigen-presenting cells, meaning 3 types of white blood cells. Will take a pathogen, so take an invading bacteria, for instance, engulf it through phagocytosis, break it down, and what these cells will do is they'll take a little piece of that broken down pathogen and they'll shove it onto their own surface with these receptors called MHC, these little receptors that are like little hands holding out the broken up corpse of that pathogen, okay? So that your white blood cells can now see that's the bad guy, okay? So antigen presenting cells will hold out a piece of the pathogen to tell the rest of your white blood cells, this is the guy I want you to go after. Now, the 3 types of antigen presenting cells, the way I remember it is DMB. I think of kind of like hearing the DMV, which no one likes. So DMB, dendritic cells, macrophages, and B cells are the 3 types of antigen presenting cells, okay? So dendritic cells, macrophages, and B cells. And again, they do this by MHC, specialized proteins, which are those little hand-like receptors to hold out the piece of the pathogen. Here I wrote out those 3 antigen presenting cells so that you know how they are spelled. Now I keep saying B cells and T cells, so I threw in this slide so that you can really see what we mean by B cells and T cells. First of all, they are named based on where in your body they mature. So B cells are called B for bone marrow because that's where they mature into the cells that you need. And then T cells mature in the thymus. Now each of these cells has a significance in your adaptive immunity. B cells are important because they are what turn into the cells that secrete antibodies. So all of the antibodies in your body have been secreted by B cells. Again, you see it written as plasma cells, but that's just the differentiated form of B cells. B cells are also important because you saw on the previous slide they were listed as one of the 3 antigen-presenting cells. T cells then are the real heavy hitter fighters of your adaptive immunity. They include the cytotoxic T cells, and again, when you see cytotoxic, that means they're killing cells. So cytotoxic T cells will physically kill the pathogen cells as well as any of your infected cells. You have helper T cells that basically help activate and start up the adaptive process. And then you have regulatory T cells, which are the cleanup crew. Regulatory T cells, after your body has finished fighting an infection, they basically shut down, suppress your immune response, and clean up the mess. Now, when we keep saying that adaptive immunity involving B cells and T cells gives you memory immunity, meaning you can't get sick twice. Again, I explained that before, but I'm going to keep using it as that phrase. So that memory is because in this response, some of your B cells and T cells that have fought this infection are turned into memory cells, meaning they stay in your body for future infections. So if you encounter that bad guy again, those memory cells can quickly get rid of it. Now here I'm going to summarize what we actually mean by the adaptive immunity response. And again, this is a concept where if you look online, if you watch videos, if you read textbooks, it is highly complicated and crazy the way they write it out. Here, I stripped down all the extra details and just summarize for you the key points of what your adaptive immune response actually is. And I want you to remember that adaptive immunity includes both the humoral response and the cellular response, okay? So humoral focusing on B cells and cellular focusing on T cells. So both of these are happening at the same time. Like I mentioned in the last slide, helper T cells are going to help kick things off. So what happens is one of the antigen presenting cells, either a dendritic cell, macrophage, or B cell, remember DMB, will present the antigen, meaning it shows the helper T cell who the bad guy is. It shows it which bacteria or virus is currently infecting you. That then activates the helper T cell to activate B cells. Okay, so the helper T cell, now that it knows which bad guy to fight, it stimulates the B cells to turn into those specialized plasma cells that we said produce antibodies. And then the antibodies help you fight the infection. At the same time, the helper T cell is also producing chemicals that help turn naive, meaning not differentiated T cells, so baby T cells, into full-blown cytotoxic T cells, okay? Meaning those cell-killing T cells that will basically release chemicals that trigger apoptosis of pathogen and of your infected cells. What apoptosis is, picture popping cells, okay? These cytotoxic cells trigger a burst or a murder, basically, of the bad guy cells, okay, which will either be pathogen or your infected cells. Now, at the end of this, some of those activated B cells and T cells are now kept as the memory cells to then stay in your body so that next time you encounter that same antigen, your body doesn't need to take the time to do antigen presentation, to activate the helper Ts, to activate the B cells, to activate the cytokines and the cytotoxic blah, 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 right? It doesn't have to take the time to do all of that because it already saved some of those fully formed fighting cells specific to that pathogen, okay? So again, keep in mind this is adaptive immunity, meaning it is specific. So for instance, if you are fighting COVID-19, well, what happened was that antigen in the beginning was a piece of that COVID-19 virus, of one of its proteins, that was presented to activate the B cells to produce antibodies specific to COVID-19, and then to activate the helper T cells to activate the cytotoxic T cells to specifically kill COVID-19. Okay, and now you have kept as memory cells some of those activated B cells and activated cytotoxic T cells to then identify COVID-19 the next time it comes into your body and quickly attack it. Now, there's one thing I left off of this slide. I completely forgot to write it, and think about it. What's missing here? Regulatory T cells. And what would the regulatory T cells do? They would clean up after all of this mess. Okay, they would suppress your immune response once you have fought the good fight and finished fighting that infection. Okay, so sit with this slide, repeat it to yourself. You can rewind, play it again, and maybe write out for yourself. But again, this is the condensed, summarized version of what adaptive immunity really means. Now, with immunity, there's a lot of terminology you'll encounter, but like with everything else in biology, if you just break down the words, it'll really help you figure things out. So your immunity can either be naturally or artificially acquired. And what I want you to write down is the difference between those two is whether or not a doctor and shots are involved. Okay, meaning for natural, I want you to write naturally acquired immunity means there is no doctor, no shot, no injection into your body. Whereas artificially acquired immunity, you will have a shot, you will have some sort of injection, you will have a doctor involved, okay? Now they further break that down into passive versus active immunity. And again, if you think about the terminology, it will help you figure out the difference. So if you hear the word passive, that means nothing to do, no energy, expended. You could just sit there and chill because you are given already made antibodies. Whereas active, anytime you hear active, that means energy involved. Okay, so active means that your body has to do work. It has to use energy, which in this case for immunity means it has to make antibodies from an antigen. Okay, so once you kind of distinguish those terms, it becomes easy to figure out if I give you a scenario, you should be able to tell right away, is it naturally or artificially acquired, and is it passive or active? So for instance, if I say that antibodies are passed from a mother to her baby through breastfeeding, what does that tell you? Well, first off, is that natural or artificial? Well, did she give the baby a vaccine? Is she in a doctor's office? No. It's a mother and the baby, no doctor, no shots, so it's naturally acquired, right? And then, does that baby have to make antibodies? What did I say was given? Antibodies were given, right? So it doesn't need to make antibodies from any antigen. It's already given fully formed, made antibodies. So that scenario of a mother passing the baby antibodies in breast milk, that would be naturally passive acquired immunity. Okay, so naturally acquired passive immunity. You can order the words any way you want, by the way. So just be able to recognize natural versus artificial and passive versus active. So I'm going to give you another example. Let's say you go to the doctor and get an injection of antigens, meaning bacterial proteins or viral proteins. Okay? What kind of immunity did you just get? Send me a remind message and let me know if you go to a doctor's office and get an injection of antigens, pieces of a pathogen. What kind of immunity did you get? Was it naturally or artificially acquired, and was it passive or active? So when you get to this slide, send me a remind message for getting an injection of antigens. Now we keep mentioning antibodies. Well, what exactly are antibodies? Well, as you see here, they're a Y-shaped piece of protein, and on their edges they have what are called antigen binding sites that come in all different shapes and sizes to specifically bind their matching antigen. Please keep in mind, antigens are the protein name tags from the pathogen. So these antigens are part of cells, whereas antibodies are what your body produces that can then bind to the antigen. And once they bind together, you then end up having a reaction occur that can help protect you. Now, the way that binding can protect you, there are a few different examples. Here are 3 ways that antibodies can help you once they bind to that antigen. There's neutralization, opsonization, and complement activation. And one of those should sound familiar to you now that you're experts on it from earlier slides. So first up, we have neutralization. Basically, what that means is that the antibodies will bind to the pathogen. And by coating that pathogen, sorry, I thought I had the laser pointer on. By coating that outside of the pathogen, it's now making it tougher for that virus or bacteria or toxin to then stick or attach to your body, your tissues, or your cells, okay? And attachment is a big part of how pathogens hurt you. So neutralization means antibodies can coat the outside of a pathogen and prevent that pathogen from binding to your cells or your tissues. There's also opsonization, meaning that when antibodies bind to the outside of a pathogen, that now can tag that pathogen to get targeted by a phagocytic white blood cell. So usually the big bad macrophages, okay? But any phagocytic white blood cell will now recognize that antibody-coated pathogen as something it needs to engulf, chew up, and digest down. The third way that antibodies can help you is by activating the complement system, and you are already pros with the complement system. What does that mean? These antibodies can activate the proteins that then drill holes in that pathogen, killing the pathogen. Now you'll see some terminology with antibodies. It includes affinity, avidity, and cross-reactivity. What affinity means is basically how well an antibody will bind to an antigen, okay? Your affinity for something or an antibody's affinity for an antigen means how strongly does it like it? How strongly is it attracted to it? Okay, so if that antibody has a high affinity for a particular antigen, it means it will strongly bind, it'll really attract to it. Low affinity means, the antibody won't really look to bind to that particular antigen. Okay, so affinity is how strongly attracted the antibody is to the antigen, whereas avidity basically means the strength of all of the antibodies binding to antigens at once. So basically meaning that avidity is higher if there's more antibodies that will bind to a particular pathogen or structure. Okay, so avidity means higher numbers, more antibodies. Lastly is cross-reactivity, and that basically means that there are sometimes different epitopes to antibodies and antigens, meaning slightly different shapes. Okay, so sometimes cross-reactivity means that the same antibody might be able to bind or interact with a few similar antigens rather than just one specific antigen. Okay, so I'm going to type out some definitions of these just in case you missed what I was saying, so you can kind of see a clear written out form. So here are the definitions for you to pause the video and jot them down. Now, the last thing I want to point out is that, even though your immune system is trying to help you, sometimes it can end up hurting you or working against you. And the three big examples for this are tissue and organ rejections, autoimmune diseases, and allergies. So with organ rejection and organ donations, the problem with this is when we covered your blood in a previous lesson, you saw that all of your blood cells have antigens on them, whether it's type A or type B. And in your body, you also have antibodies against anything that's not yourself, that's not your matching blood. So what can happen is if you are given tissues, organs, or blood from someone else who does not have the proper, self-matching blood type as you, well, now your antibodies that attack the invader will bind to that foreign blood or tissue's antigens. And when antibodies bind to antigens, that's called agglutination, which is a fancy way of saying they clump up. Okay, they clump up and they can cause clotting and complications throughout your body. You do not want all of your blood starting to clump up. Okay, what can help, however, is if they give you immune suppressing drugs when they do any kind of procedures to help block any complications and wrongful clotting. You can also have autoimmune diseases, and what autoimmune diseases are is what they sound like. Your body is now attacking its own cells. Okay, it thinks that these cells or antigens that are actually you are foreign invaders. The last type of problem that your immune system can cause are allergies, and in science we refer to those as hypersensitivities. So when you see hypersensitivity, that tells you we're talking about allergies. And these allergies can either be immediate, meaning right away you start itching, getting swelling, going, I can't breathe. Or they can be delayed, meaning you may, for instance, touch poison ivy today and you won't see a reaction until 4 to 7 days later. Okay, and we'll talk about in a minute why that is. Now, I just want to give you a little more information about a few of those immune issues that we just mentioned. The first one is called type 1 anaphylactic hypersensitivity. And I know that sounds like a mouthful and big scary words. That's a fancy way of saying immediate allergies. Okay, so this means you start having an allergic reaction to an antigen, which we call it an allergen when it's specifically causing a hypersensitivity, and you have it right away. Now, some examples of this would be, for instance, your allergies to pollen or to cats or to dogs, any of those things where very quickly, peanuts is another big common allergy, where you right away, as soon as you encounter that pollen or that dog hair, you start sneezing and sniffling and have trouble breathing and coughing and all of that, because it's immediate. It's type 1 anaphylactic hypersensitivity. Now, what's scary about these kinds is they can get very problematic for you. Now, the reason why you feel the symptoms you do with allergies is because they release what's called histamine, leukotrienes, and prostaglandin. And you already know what histamine does. That's a vasodilator, so it's opening up that blood, causing inflammation. Leukotrienes, what they do is they cause prolonged contraction of your smooth muscles. So for instance, contraction of smooth muscles, if you think of when you have a really bad allergy and you feel like your throat's closing off, that's because of leukotrienes. There's also prostaglandins, which can further affect smooth muscles, but they're most famous for increasing mucus secretion. So when you think of these three things, I want you to know histamine is a vasodilator, leukotrienes contract your smooth muscles, and prostaglandins will cause mucus secretion. So that's why you start going when you have allergies to things like dogs, cats, pollen. Now, these reactions can either be local, meaning they stay in one place, such as a patch of hives on your leg or on your arm, or they can be systemic, meaning it goes towards your whole body, and that usually can trigger what's called anaphylactic shock or anaphylaxis. And that could be deadly because it can end up causing your circulatory collapse or respiratory system collapse. And so that can cause death from a simple, allergy, okay? The alternative, another type of allergy, is called type 4 delayed cell-mediated hypersensitivity. And that's a fancy way of saying delayed allergies, okay? So know that type 4 is delayed, whereas type 1 is immediate. The reason why this is called a delayed allergy is because it relies on T cells. So the reason why you have that immune response of things like inflammation and redness, swelling, itchiness is from the activation of T cells. Now, as you know, T cells and adaptive immunity is very slow. It's a series of activations, right? Helper T cells got to activate this cell, got to activate that cell, and make memory cells and all this other stuff. It takes time. So that's why when you have something like poison ivy or allergies to what they call contact dermatitis, so allergies to basically certain types of metals or certain jewelries will cause a rash, it's type 4 delayed because it's dependent on T cells, and that takes time. The adaptive immunity takes time. So for this, I want you to especially know the example of poison ivy. So if you get poison ivy rash, that's contact dermatitis. That is a type 4 delayed hypersensitivity. This is just a summary of some of the information to show you the difference between immediate and delayed responses. I want you to circle immediate response, and above that write type 1 anaphylactic hypersensitivity, whereas delayed, circle that and write type 4 cell-mediated hypersensitivity so that you remember the difference between them. Now, the last point I want to make is about autoimmune diseases. And autoimmune diseases are very problematic nowadays, and they still don't fully understand a lot of them. So it can be brutal for the people who are fighting them. It can take them years, even decades, before they get any help or diagnosis. Basically, what autoimmune disease is your loss of self-tolerance. And yeah, I know it sounds funny. Yeah, I cannot tolerate myself. No one can. But what we actually mean by loss of self-tolerance is that your body, your immune system, no longer sees your own cells as yourself. Instead, it loses the ability to distinguish self from non-self, so it starts attacking and damaging your own cells and your own organs. And some big examples of this that I want you to remember are lupus, multiple sclerosis, and rheumatoid arthritis. And I always emphasize rheumatoid arthritis because a lot of people, when they hear anything with arthritis in the name, including rheumatoid arthritis, they brush it off as just, oh, it's just old people, they can barely move their hands, that's it. That's not what rheumatoid arthritis is. You can get it as a 20-year-old. It is your autoimmune system, your own body attacking all of your joints, and it can end up being deadly once it starts hitting your respiratory system. So please keep in mind that when you hear lupus, multiple sclerosis, or rheumatoid arthritis, these are all examples of autoimmune disease. And there are many different causes of autoimmune disease, things like genetics and toxins. They don't fully understand everything yet. Like I said, they're still studying them, but they can be very brutal. Now that is it for this chapter, and that's the last chapter of week 2. So please make sure that you add this material to your study guide, that you upload your study guide by Friday, and that your studying guide includes all of this week's material. Also, since this is the end of material for week 2, please make sure you also do your discussion board posts by Friday as well. Okay, as always, any questions, any concerns, send me a message and remind day or night. Thank you and have a wonderful day.